All right, I think we will go ahead and begin. So welcome, everybody. Welcome to the Release 60 webinar. My name is Evelina, and today I will be presenting together with Sam Rand and also Suzanne Morenfeld. So just a few housekeeping things before we begin. In case this is the first time that you're joining us, we are using the GoToWebinar platform, and we are happy to accept and answer your questions during the webinar. If you would like to ask any questions, please type them in within the questions section within the GoToWebinar platform. You may have slightly different look depending how you're joining the webinar, but we will try to answer as many questions as we can during webinar live, and we'll get back to you with anything that requires a little bit more information. So in terms of the recording, the webinar is being recorded and will be posted on our YouTube channel within 24 to 48 hours. We will also send you a link in the follow-up email, as well as any certificates for your participation. Right. So let's go ahead and begin. Today we will talk about the new features and new datasets that we have released in VEuPathDB Genomics site in this release. I'm on the main page for the VEuPathDB.org project, where you can access the updates about the recent datasets and features within our news section. I will demo some of the updates within the FungiDB website, but these updates apply essentially across all our genomics site. If you ever have questions that will come up, for example, after the webinar, please feel free to contact us by clicking on the Contact Us link at the top of any of the genomics site, and it will take you to the form that will allow you to send us a message, and we will try to get back to you as soon as we can. Right. Now I will navigate to the FungiDB website. So this is the main landing page, and I'll quickly navigate to the news section. In terms of the new features that we have released is an update to our dataset record pages that allows you to identify common datasets or datasets that are linked via publication. For example, if you want to learn more about how to find these datasets, we have a very nice tutorial using GRDADB as an example. And I will also show you how you can find these datasets in FungiDB. For example, if I'm on FungiDB and I know that there is a dataset that we have integrated recently and that had investigated the transcriptomes of both Candida and human cells when Candida albicans and other species were incubated with whole human blood, I can now identify all of these datasets across different databases in VEuPathDB. And I can do this by simply scrolling down the dataset record page and navigating to the associated datasets section. Within this section, it's already expanded, but if you expand it, if it's closed by default on your page, you will be able to see that a single publication is associated with all of these datasets in two different databases, FungiDB that integrates fungal and oomycete species data, and also HostDB that integrates datasets specifically for hosts. The really cool thing about this that now if you're interested, let's say in Candida response to growing with human cells, now you can navigate to individual datasets in fungi and investigate the transcriptomes of these species. And so the transcriptomes were taken across several time series. And you can also navigate to HostDB. And so if you click on one of these links, it will take you to the corresponding dataset record page in HostDB. So you can essentially set up the same searches, RNA-seq evidence searches in HostDB, mining the expression data in human cells. Now, this is a quick update about the the linked dataset. And so datasets, and now I'll go back to FindGDB main news section and walk you through in brief about the updates that we have had in this release. Now, in terms of the data, we have updated several genomes, and this included minor updates to avoid discrepancies and sync the records between INSDC versions and also VEuPathDB. And we have integrated a number of genomes for model systems and also human and plant pathogens. For example, we have human pathogens such as histoplasma. We also have Candida and Aspergillus and Coccidioides. For Coccidioides basidiaceae, this is a new annotation, in-depth annotation and re-annotation of Silveris strain that was integrated in parallel with the original Silvera annotation, so that you can bring this genome up in Genome Browser and essentially look at the improvements that have been made compared to the older genome. We have also integrated a number of SNP records, and these indicate records for human pathogens such as Aspergillus fumigatus, Candida auris, which is new and emerging pathogen, and also plant pathogens. For example, if you're interested in human pathogens, you can mine these this data record set by navigating to the dataset record page and examining the type of the searches that are available. For example, you can set up a search that uses SNP characteristic search to identify genes with non-synonymous mutations. And here's Candida auris already pre-selected, as I have pre-run this search, and so my selections are being remembered. And the data set that I'm interested in is the 2 isolates from the Netherlands, essentially the original isolates detected for the first time in Netherlands. And so what you can do is you can run various types of searches, including non-synonymous mutation search, to highlight any mutations that may be interesting, for example, in terms of growing the antifungal resistance of these pathogens towards various drugs. As we know, non-synonymous mutations are the #1 contributors to that. So in this case, I have already pre-run my search, and here's the result of this search strategy, where a first step essentially can be a step to a much detailed strategy involving RNA-seq records and other types of records within FindGDB. If you scroll down at the bottom of the screen, you'll have a summary of the results that returns essentially over 5,000 genes that have non-synonymous mutations, and also tells you their positions, their gene IDs, and etc. The cool thing about this, that you can visualize these records not only within the search output such as results table, but also within JBrowse, Genome Browser. Here, for example, I am viewing the genome for Candida auris. I also chose to activate combined RNA-seq plot, which uses the information from all RNA-seq datasets currently available in FindGDB, and also displays records such as SNPs by coding potential. Now, the SNPs are already shown, highlighted in terms of their features, such as the coding effect that they have. For example, red will be stop or nonsense mutations. Dark blue diamonds will be showing non-synonymous mutations, and you have synonymous, and also in yellow will be highlighted non-coding mutations. And if you're interested to learn a little bit more about the specific isolates that we just used to run our search in the SNP dataset records, you can zoom in further and bring in coverage and alignment tracks for the 2 isolates that were sequenced in the Netherlands dataset. Right, now I will come back to, again, FindGDB and look at the news section again. to show you some other data sets that we have integrated. And so notice that I have mentioned obviously just one, but we have a lot more data, especially coming in for Aspergillus fumigatus, which is the leading pathogen of aspergillosis, an invasive disease in humans. We also have a number of transcriptomics data across different systems or models that includes models for filamentous fungi such as Neurospora crassa, also human pathogens and plant pathogens that pose food security risks. Now, the transcriptomics data can be mined essentially by clicking on the dataset. For example, here I will use an example dataset for Cryptococcus neoformans. This dataset is the last of the series that we integrate, where the authors have taken a in-depth approach to studying transcriptomics in both KN99 and also JC21 isolates of Cryptococcus neoformans, and try to define not only detailed transcriptomic profiles when they're grown under different conditions, but also define the positioning of the 3' and 5' ends. And so one of these data sets sort of accumulation of the integration of these series, and you can explore this data set by navigating to the dataset record page and essentially scrolling down, looking at the type of the searches that we have available. For example, you can look for differentially expressed genes and use the criteria to select your reference, such as you could use stationary phase growth as a control, and you can use the exponential growth as your comparator sample and look for up and down regulated genes. And so this is just a quick summary of how you can set up your search, but imagine that you can modify it to fit your specific interests. And once again, I'll come back to the news section and talk a little bit more about the other types of data that we have. Now, we also have integrated genome browser tracks where authors have used RiboSeq to define the binding sites within the Candida albicans genome. We also have lots of phenotype data integrated in this release, and that includes manually created phenotypes. So for manually created phenotypes, you can, if you click on the link here, you can navigate specifically to the dataset record page that tells you a little bit more about the work that we do. The manually curated phenotypes are essentially those that we curate manually from the publications and provider data. And in this release, we have lots of records coming in for Aspergillus fumigatus and also Pyricolaria oryzae. In terms of Aspergillus fumigatus, we have the curation annotation team curated several publications, and I created new phenotypic mappings for 47 genes. We have also integrated records from the Magna Genes database, and Magnaporther, or also Pyricolaria oryzae, is a major threat to global security in terms of the rice crops, and we have integrated the manual annotation, manual curation records from the database that was recently released. And that work was conducted by Andy Foster, Nick Talbot, and others. And both of curated phenotypes for Aspergillus fumigatus and Magnaporther were curated with specific VEuPathDB ontologies that help us to create searchable data sets, where you can come in and search for specific terms to identify genes that fall within this criteria, rather than just going through the Excel spreadsheets that contain text. And finally, the updated annotations, such as summary of annotation updates and etc., as well as GFF files, can be also found within this section. So in a few seconds, I'll pass it on to Sam, who will talk about updates for the vectors of human disease. And after that, Suzanne will also pick up on the updates for other genomics sites, and she will also discuss some of the other updates we have had within the release 60. So Sam, I will give it, I'll give the floor to you.
Thank you, Eve. Just need to share my screen. Oh, oh gosh.
I will make you a presenter. Give me just a second.
Actually, Suzanne, can you go first? I'm gonna have to restart. I'm sorry.
Sure. I can.
I apologize.
Oh, no problem.
That's okay.
So, can everybody see the VEuPathDB home page here?
Yes.
Okay, cool. So, you know, the parasite databases received a lot of good data in release 60, and I'm going to go through an overview of those and then go into more depth about an RNA-seq data set that's in VEuPathDB. And the news is the best place to get an overview of what's in release. And so I'm in VEuPathDB, which supports all of our organisms. And if I open the news, then I'll be able to see, I'll be able to scroll through site by site. There's VectorBase and then TriggerDB. But before I go into the organism-specific data, there are two data-related items that I'd like to mention, since these were integrated across into VEuPathDB across the board. And the first one, now I say across the board, but that's not exactly true because VectorBase and HostDB did not have a problem with pseudogene classification. But on every other site, we did have, we had fixed something and we wanted you to know about it. We've corrected an issue with our pseudogene representation. So due to an incorrect pipeline that has been corrected, the pseudogenes that had an internal stop codon automatically receive prime UTRs, which is incorrect. And we've corrected that, and now all pseudogenes are displayed without coding sequence and UTRs, and the biotype is pseudogene. So here's a, if you're interested to see, if you're looking at pseudogenes, if you're interested to see what genes have changed, there's a search here that's linked to this in every site. And that takes a while to open, so I've actually opened it in another screen. And there's about 56,000 pseudogenes that were corrected. And so if we go to one of those, I chose just an ID at random, and that ID happened to be this unspecified product in Microsporidia DB. And it is classified as a pseudogene. When I look at the gene model, I can see that it is white, which means that it is not a protein coding gene, and it does not have and it does not have a UTR. The other thing that is notable about pseudogenes is that we do not add the protein sequence to the sequences section. So all of our pseudogenes are now correctly represented. And so I'm going to go back to the VEuPathDB of the news. And the other thing that we did that is across actually all of our sites, right, is that we have updated the AlphaFold data on all of our sites. I'm going to go to the AlphaFold homepage, and you know, AlphaFold is a protein structure database. They predict protein structures from the sequence, and then those predictions are made available through a partnership with ENVIL and EBI. So we have downloaded AlphaFold Release 4 and associated their predictions to genes in our system using the VEuPathDB. Their Release 4 contains over 200 million proteins, which is about 100-fold more than previous. So now, if you look in our sites, we have about 4 million genes that have on their gene pages. So let's, there's a search on every site that returns genes with AlphaFold data. Let's go there. I'll go to VEuPathDB, and it is under search for genes based on structure analysis and AlphaFold predictions. And here we are on the search page. I'm going to select all organisms and say get answer. That search returns 120,000 genes that have AlphaFold predictions on their gene page. Go ahead and choose one and we can look at it. We can click on structure analysis here. I'll open it up and you'll be able to find AlphaFold. I can also use this. Go to the AlphaFold section, and the AlphaFold on the gene page is actually in two sections, one called AlphaFold. And another with the prediction visualization. The first is just a table, and this link will take you to this gene on the AlphaFold site. We have some other data associated with that in the table. And then we have also integrated this page. We've integrated their plugin for the AlphaFold visualization software so that you can do a lot straight from this gene page. And here it is. It just didn't load properly the first time the gene page opened. So, so this second AlphaFold section on the gene page contains this plugin where, you know, there's lots of unique features here, right? So I can scroll over the sequence and then see that moving into in the protein and vice versa. I can look at that. I can move this around. There's look at details down here in the corner. Details appear as I hover over each amino acid. So there's a lot you can do straight away from the gene page. And there are about 4 million genes across VEuPathDB that have this data now on their gene pages. So now I'll go to back to VEuPathDB and look at the organism-specific data. And I'll go through VectorBase. Well, Sam is going to talk about VectorBase, so I might just point out that the new data in VectorBase has a couple of genome updates, some sequence 3 new sequences, some proteomics, RNA-seq, and of course the AlphaFold predictions. And Sam will talk about the population biology data. And I can continue to scroll through all of the sites, but that tends to give me a headache. So I've created this, you know, this sheet here that just has a summary of everything. So the first, this is all organized by data type, and we have genome updates, and all of these sites, VectorBase, TriTrip, Toxoplasma, Puro, and VEuPathDB, received an update to at least one of the assemblies. And so I'd like to give a little bit of background about that. So some of our genomes were given to us directly from the researcher and were never deposited at a repository such as NCBI or EBI. And then some other data we downloaded from the repositories, but then the researchers have updated that assembly at the repository. So in the past year or so, we've tried to sort this out and minimize the number of public versions that are available. So that's what these genome updates are. We've synced our version on our repository with the repository version. In some cases, we've actually uploaded our version to the repository if it was missing from there. We'll be seeing genome updates from then on. And then in release 60, these sites received new annotated genomes. These are new organisms that our site now represents. The TrickDB received 2 histobonus genomes, annotated genomes, and one of these is an attenuated strain and the other is a virulent strain, so that might be really interesting to look at. And then MicroSporidiaDB received quite a few new annotated. Then other functional data that went into the parasite databases is VectorBase received a proteome data set and RNA-seq data sets for integrated into vector-based host and AmoebaDB. And we can't forget about our annotators, right? Tritrip, Plasmo, and FungiUB all have curators assigned to certain organisms there, and we've improved the functional annotation with, if you go to the news, you'll find tables there and access to what genes have changed and the improvements made in the annotation. I believe Eve showed you that. And then where there are new user comments placed on genes, those have been integrated and reviewed. So that is an overview of the data across the sites. And I'd like to go into depth just to demonstrate the RNA sequence data that was in AmoebaDB. Now, the thing is that I'll show this, amoeba-specific data set, but if the features are exactly the same across all of our sites. So if you're a fungi specialist or a Plasmodium specialist, you could go to any that you have the same searches and features available on that site. So for I'm going to take you to the data set a little bit different way. Instead of going to the news, right? I'm going to go to this RNA-seq search page, and I'll say search for genes based on transcriptomics and RNA sequence. And then I'm at the search page, and these are all data sets, most of them published, that I think all of these are published, that we've integrated. So that means that we have gone to the repository, we download the raw sequence data, and then we run our own RNA-seq workflow according to that. And here, so our analysis is a little bit different than the published method, but that's a good thing actually, because all of these data sets are now comparable. They've all been analyzed by the same workflow. Okay, so this is the new one from release 60. And if I hover over this question mark, I get a summary of the data set and a link to the publication. So if I open that up, I can get to the PubMed abstract, and then I can also open the publication. If I want to look at the figures, I can do that here and use image. Okay, so let's talk a little bit about their experimental design. They started out with a trophozoite, a Castellani strain F, and they incubated that under encysting conditions and regular control conditions. And they did that for 8 hours and took time points at 1, 4, and 8 hours during encystation and 1, 4, and 8 hours control. Then they isolated RNA, protein, and phosphopeptides. Now we've integrated in release 60 the RNA. We have the proteomics data, but that's been delayed a little bit. It'll come out in a future release. For the RNA sequence data, they analyzed their RNA according to RNA sequence, and then they looked at differential expression and functional annotation and Go enrichment. So let's see how we can do the same thing, more or less, with VEuPathDB. So I'm going to go back to the search page, and I'll notice that this has 3 searches available: the differential expression, the fold change, and the percentile. If I open up the differential expression, then I see I actually have pages here for each of the searches, right? So for differential expression, I'm going to look at, differential expression, because they had triplicates at each of their time points, that sets it up nicely for statistical analysis. And so this differential expression search accommodates, right? So I'm going to compare the encysting 8-hour time point to the control 8-hour time point, looking for upregulated genes with a fold difference of 4 and a p-value of 0.0001, so quite stringent. And if I say get answer, then I am returned 1,098 genes that were upregulated during the encystation. Over here on the left is the organism filter. I want to expand all. I can see that only Acanthamoeba castellani strain F is represented in this gene result, and that's because the RNA-seq was aligned to this genome. Right? There are no other genes that contain this data. So if I close that, you can go to these gene pages and specifically look at transcriptomics. We have a summary graph that compares the values for this gene in this experiment compared to others that are integrated in VEuPathDB. And then you can also look at specific details of each experiment here in this table called transcript expression. The other thing you can do with this, which they did in their paper, was to run a Go enrichment. So, and that's available just a couple clicks away. I'm back here at the gene result of these 1,000 or so genes, and I click Analyze Result, which takes me to our enrichment tool. And I'm going to choose Go ontology, although I can look for metabolic pathways or word enrichments in certain fields associated with those genes. So for the gene ontology enrichment, I'm going to choose the molecular function ontology, and then just submit that. And what it's doing is running a Fisher's exact test of this group of genes, these 1,098 genes, versus the entire genome for Castellani F. And it was pretty quick. And what I see is, a list of Go terms that were significantly enriched in my 1,000 genes. And the first one is GTPase activity, which was enriched, has a p-value of 10 to the minus 12 and a corrected p-value of 10 to the minus 5. So quite a significant finding. And it's available at your fingertips if you'd like to. You know, okay, so other things we can do with this data set now that we're in the strategy system is to say, say we want to look for genes that have also transmembrane domains, right? So I can run that search, look for any gene on the site that has a transmembrane domain, and I find that 200 of those 1,000 genes have transmembrane domains. Okay, so let's go back to the search, and I'll show you what the fold change search does. So first we ran the differential expression search, now we run the fold change. And the thing is, even though this experimental design included replicates and you could look at statistics, it can be a little bit limiting. So we have this fold change search so you can really explore the data. Where you could group samples that you know, in a differential expression search would not be groupable because of the statistical analysis, the t-test that we did. So I'm going to say compare all of these insisting time points to all of these control time points and find me genes that are upregulated by a fold 2. And there's some other parameters that you can set if you want to get into the nitty-gritty, but I'll just leave them at default and I'll say get in. So in that way, I've found almost 5,000 genes. Actually, let's go ahead and revise this to be fourfold. I think that's what we looked at before, and say revise. We actually come up with about 600 more genes here. So this is a fold change where you can explore different groups of data of samples, whereas this is a little bit more limited, but is backed up by statistical analysis. So then you also have, you know, the Go analysis available, Go enrichment. Go ahead and run that. How that differs. Okay. Right. So again, we have the GTPase activity. At a good 10 to the minus 10 corrected p-value. So there is one more search available for this data set, and that is the percentile search. So what we have done is,
Zen,
you know, we've read.
just a quick question before you move on. There's a question about, can you go a little bit more into the interpretation of Go function results? So maybe we could just take a small pause here before we move on, if that's okay.
I can try. Yes. So the gene ontology is a controlled vocabulary that is used across all organisms, right? So somebody has sequenced this organism, they've assembled it, they have called genes, right? And so say somebody does that in mouse, right? We have the mouse genome annotated, and we have the, you know, the acanthamoeba genome annotated. There are certain genes, right? There's only a certain number of functions in a biological organism, but and the gene ontology was created so that similar genes in the mouse organism could be related to similar genes in the other organisms without them being orthologs or related by structure and all that kind of stuff. So curators, actual human beings, either using a computer program or by manual curation, will go in and assign labels from this ontology, names from this ontology to individual genes, right? And those ontologies have to do with, there's 3 ontologies, one that has to do with biological process, another for cellular component, you know, where the gene is located, and by molecular function. So if you look at that as a whole across, you know, within Castellani-Neff, you can actually say, well, this group of genes has a certain list of Go terms assigned to it, but the background, which is the entire genome, has this list. It's a much bigger list. And so you can compare the list, this 1,700 genes versus the, I don't know, 7,000 that are in the whole genome, and say, are any more present more often to a larger percentile in this subgroup as it is. And that's a statistical analysis called a Fisher's exact test, I believe. And that's what we run when you click submit. And then the results are this Go term was found 78 times in my 1,700 genes, but it occurs 321 times in the genome, all genes from the genome. And so that translates, after the statistics, to a fold enrichment of 2.3 and a p-value, an adjusted p-value of 3 × 10⁻⁻10, which is quite significant. So it's a way to assign functional characteristics to this group of genes. You know they're differentially expressed, but now you also know that GTPase activity is upregulated in the insisting samples versus the control samples.
Right, thank you. And I guess there's also one caveat with this type of analysis. Obviously, any hypothetical or poorly studied genes may not have any Go term annotations. And as such, when you run enrichment, let's say on the 1764 genes, you also need to be aware of how many of those genes are actually hypothetical and subtract them from, you know, the Go enrichment ontology terms that you'll return, assuming that they're not present simply in that group. You can run an additional search to identify any genes that fit within the criteria if you wanted to go and look at them a little bit further, and let's say look for any domain signatures or motifs, etc., and see if you can learn a little bit more. I see there's another question. If you click on the gene results, they will probably have common genes with different categories. I'm assuming that you're talking about Go ontology, Go terms. Let's see if you click on the gene results, they will probably have common genes with different categories. So yes, if I misunderstand you, if you could just put a little bit more details in the chat, we can get back to it. But yeah, Go terms annotated across different genes and different genomes. So you can envision that the same Go term can be applied to multiple genes within the cell. And what Suzanne is showing right now, she is actually showing one of the hypothetical genes that I have mentioned that probably will not be returned in the Go enrichment analysis. Okay, thank you, Suzanne.
Just one tiny other thing. I'll let I'll just mention that this percentile search is, but we analyze this according to our RNA-seq workflow, and then we have values, expression values for all of these samples, and then we rank them from high to low within a sample. So we take all of the values, all of the genes that have values in this 61-hour sample, we rank them from the highest expression to the lowest expression, and then break that into percentile groups. Like here's the top 99%, here's the top 90th percentile, and then we let you go and search based on that. So let's say we want to look in the cysteine, we want to know what genes are expressed, are highly expressed in that. And this is good to add any strategy that you want. Okay, that's all I want. Sam, are you ready?
Yes.
Okay, I will send it to you, I think.
Show my screen. Okay. Can you all see vector base? No, you can't. Okay.
We're seeing Slack. Yeah.
That's okay.
Mm-hmm.
We can do it this way. Sorry about that earlier start. So yes, I'm going to be speaking today about some vector-based map view projects specifically. So I'm here at the vector-based website, and I too will go to the vector-based specific news. And I will be talking about these 5 population biology data sets. Now, when we say population biology, we mean that it appears in our map view portion of our website, which is only available in vector-based right now, not the other component sites. But if we get data sets that are relevant, so if you work for something else and you think there's a data set that would be relevant to our map view, please let us know. And what do I mean by map view if you haven't been? So I'm going to click on this first data set view on map. And what I see here is we have our map, and I can zoom in. And this is from the Rhode Island Department of Health. And we see here, this is abundance data. So I'm going to click on our abundance view, and it'll switch a little. And I can click optimize color to see some prettier colors. And so here we have a regular trap level data on the population of mosquitoes that were collected. And what I mean by trap level is I keep clicking in until we get down to the individual traps collections over time. Sorry, just because of the window. Okay, sorry, my notes. The next data set is from the is some tick data, and we've been doing a lot of work on collecting, making a special effort to curate both tick biodata and soon many genomes. So here, for example, is a lot of data that we have specifically on Ixodes scapularis from across the eastern half of the United States. But I'm specifically going to talk about one data set, which we see here, where again we have this that abundance style data that I was telling you about. And this is from the Tran lab at the University of Pennsylvania, but it's over 500 different sampling locations, and they performed, I'll click on a data set, and we have these different views on the left, so I'll click on this timeline view, and we can see, and I can increase the resolution. We can see the number of Ixodes scapularis ticks that were collected from each of these 500 plus sampling locations across the state of New York over time. And as you can see in map view, we work really hard to standardize and get very detailed metadata. So you can see for any of those collections, we know when it was collected, how many, the sex, if known, the developmental stage, how it was identified here morphologically, and of course the where. So we know the where, the when, the who, etc. So we really encourage you, if you're doing maybe seasonality research or information like that, that you take a look at this new data set. And I'll just say that going forward in the next release or two, we have a lot more state or countrywide presence-absence studies that have been aggregated by people from the CDC. And so just keep coming back to VectorBase and you'll see more and more tick data appearing over the next year. Next data set is an insecticide resistance data set from Myanmar or Burma. And here we have a sampling where they basically exposed many different local vectors. So you can see from one place, many different Anopheles vectors, and performed susceptibility assays using WHO paper kits and delta method. And for insecticide resistance assays, you know that the kind of metadata that you'll see will be a little different. You want to know what kind of insecticide, what kind of test, what was the concentration of the insecticide, how long, and then what was the mortality rate. So, so this one is a very susceptible species, but we can see here we start to get this vector, which is Anopheles hieruncus, is starting to show perhaps some resistance to deltametrin. Yeah, 71%. And this data set was from Myanmar. And our next data set is a small European tick data set. And in this one, we just have he just did a survey of the presence of the species, of two different species, Ixodes rhynchus and Dermacenter reticulatus. And so depending on the study, we may be able to get more or less out of the paper when we digitize it. So here we see some species presence data. And finally, the final data set in MapView from this release is another insecticide resistance assay. And here you'll notice that if I use this right-hand toolbox to explore the data, all the studies, instead of doing one insecticide on lots of vectors, we have one vector, Anopheles gambiae sensulatu, and they exposed it to multiple different insecticides to identify the resistance. And again, we can go to this tabular view and see when the collection was performed, which insecticide, what concentration, how long, what protocol, and what the mortality rate. So for example, we see that it was completely susceptible to deltametrin, but it looks like this collection in this area is kind of actually odd. Oh, here we go. We have a 60% mortality to alpha cypermethrin. So we are seeing a little bit of resistance from this location. And for any of our data in the map view, we have the different views again, and tools on the left. So you can, for example, download the data. And so I could download all the data that matches what I see visible on the screen right now. And you can get an idea of the different columns that will be in that CSV by clicking with the following fields. So you can see that we really have standardized this data and made it easy for reuse. You can share the link. So if you wanted to email someone, you see something cool, whatever, approximately what they see on the screen right now, it's been copied to my clipboard, so I can paste that in an email or a message. Yeah, so this is our map view, and please stand by. There'll be lots more tick data released, for example, with the next release, I believe. And I did want to go back and mention one thing to the first data set that I released, or I mentioned. So this is Rhode Island. So some of our mosquito abatement districts, every year or two, they give us an update. So right now I've filtered just on the most recent data set from that provider. But if I remove that, so I'm just looking at the data sets from the area, I can see that the data goes back much farther when we put together all the different years that provider has provided data. And some of our data sets actually go back 40 years of trap level weekly or daily trapping results from some of the mosquito abatement districts. So we're happy to answer any questions you have about that. You can always email us, and if you have any data that you think would look good on MapView, please just let us know. Suzanne, or you?
Oh, okay.
All right, sounds
yeah. You're welcome to.
all right. Thank you, Sim, very much. Suzanne, do you have anything else to show?
No, I think I'm good.
Okay, sounds good. So we have just a few minutes before the end of the webinar. So I just wanted to point out to people who are new to VEuPathDB sites that what I'm showing right now is the main landing page for the VEuPathDB.org. If you're interested in learning more about the various types of the searches or tools that we have, you can look within the tutorials and exercises section at the bottom. These are more sort of extensive training materials. We also have overview of resources and tools that, if you expand the window a little bit more, allow you to sort of start with the resource very quickly by giving you a summary overview of how you can utilize one or the other type of search. And if you even want to go even further and learn more, click on the help and navigate to learn how to use VEuPathDB. So within this section, you can check on the events that we have coming up and also review recordings from previous webinars. We also post materials from our workshops and other learning resources and provide other useful data that may help you during your research. Okay, and with this, I would like to say thank you to all of you for signing up and attending the webinar, and also to the instructors of this webinar, and we will see you next time. Bye. Thank you.
Bye. Thank you.